Okay, perfect. All right. Okay, is it unmuted? But I believe so. Okay, I don't want to mess this up.
It is.
Okay, great.
Yeah.
Okay. All right, good morning, everyone. Welcome to the thesis defense of Kiara Reyes Gamas. I am one of her advisors. She is co-advised by Joff Silberg and myself. Kiara is in the Systems, Synthetic, and Physical Biology program. She actually did her undergrad here at Rice in bioengineering, so you actually been here longer than I have at Rice, which just makes her an expert in all things Rice, Houston. She has very strong opinions about barbecue, but she will share with you. But before I hand it over to Kiara to tell us about her thesis research, I'll just say a few words about her. She's really passionate about environmental sustainability and synthetic biology. And I think the reason she joined both of our labs is because it allowed her to pursue both of those passions and really integrate them. So her thesis today, and this is just a word cloud of her thesis, is really focused on advancing novel tools for studying gene transfer in environmental microbial communities. Aside from her very impressive computational skills that you'll be amazed by when she tells you about her research, she's very dedicated to other things that are really critical to advancing science and having an impact. And that one great example is science communication. I searched my Slack for SciComm, and every single thing was from Kiara about events that were going on related to SciComm, workshops she's participated on during the pandemic. So this is from May 2020. She helped write a script that basically helped us translate a lot of the work we were doing on wastewater monitoring of infectious diseases to a lay audience. And I went back and read it, and I was like, wow, this is amazing. She just took these complex methods that we're using and really translated them into a lay person audience. And she's always been really amazing at kind of advancing science communication, not just in her own work, but with our entire lab. Of course, I have to mention that she's also an advocate for always making sure things are colorblind friendly. She shares her colorblind friendly palettes to make things accessible for all folks to be able to see it. And I think all of us have learned to make better graphs in that way. I also just want to highlight some of her beautiful design work. So she designed our Stadler Lab Group logo. She designed the Rice Synthetic Biology Institute logo, and she's made these amazing figures and images for our wastewater epidemiology program that I use in basically every single talk that I give. And these are lasting impacts that will, we're so thankful for the work that she's done just in these contributions in the design space. I also want to mention how passionate she is about things like responsible application of synthetic biology. She's very outspoken in her passion in this. She led a book club in our group where we discussed this book As Gods, where we talked about genetic engineering and its implications, not just in terms of scientific impact, but also economic and social responsibility surrounding this. She is not afraid to voice her opinion, in conferences. We had a conference here at Rice, and there was a representative from Ginkgo Bioworks who was on a panel, and Kiara asked a question to him directly, asking, why are you engineering bacteria and with plasmids that have antibiotic resistance genes and putting those out there in the world? And he's not afraid to put people on the spot. And I think, this really just speaks to her amazing confidence and passion for this. I have to give a few kind of quotes that group members described her as. So Kiara's outspoken, confident, and not afraid to say what she thinks. Group members have described her as someone who stands up for what she believes in, a great ally for any cause and doesn't flinch to go against the grain, and someone who says things that I think, but I'm too afraid to say. So she's been an amazing group member. Beyond her amazing scientific achievements, which she will tell you about, she's also an amazing cook and baker. She TA'd the chemistry of cooking class. She is an animal lover, has been a great cat and dog mom to many animals, and just a wonderful group member. So I'll hand it over to Kiara to tell us about her science now.
Great. Yeah, thank you so much for the wonderful introduction. Thank you all for being here. I'm really excited to share my research that I've been working on for the past few years here. It's all about making better genetic memory to understand microbial communities. So microbial communities are dynamic, complex, and they're also notoriously difficult to study. We can get a snapshot of which organisms are in a microbial community. Here, let me make sure okay, perfect. We can get a snapshot of which organisms we see in a microbial community by sequencing their genes, and we can infer what roles they play. And this can help us understand which microbes are participating in processes in the community. For example, we can find microbes that fix nitrogen in soil to feed plants. We can find microbes that are degrading contaminants in wastewater. We can find viruses that are in a community. But understanding the dynamic interactions between these organisms is really, really essential to understand these communities. Knowing which organisms interact with each other and when and under what conditions allows us to understand the fundamental mechanisms that drive processes on Earth. And there's a lot of these processes that happen in nature that we don't yet fully understand. And one of the most critical yet understudied is the movement of genes in a community through horizontal gene transfer. As a short definition, horizontal gene transfer is just the movement of genes in a population through means other than vertical descent. Basically anything that isn't cell division or reproduction. So this happens whenever organisms move genes between each other. Maybe, for example, one bacterium will give a piece of DNA to another bacterium that allows it to survive a specific antibiotic. And now both of these bacteria share DNA, even though they're not directly related to each other. Lots of organisms participate in horizontal gene transfer to get new genes that help them overcome the selective pressures that come from living in highly dynamic environments. And horizontal gene transfer is a major way that antibiotic resistance genes are spread in the environment. But it's also something that we need to keep in mind for a biocontainment context. That's because horizontal gene transfer doesn't only happen between closely related microbes, but it also happens between pretty distantly related ones too. And it doesn't just happen in bacteria and archaea either. We've seen horizontal gene transfer between two fish, that's vertebrate to vertebrate gene transfer. We've seen horizontal gene transfer between insects and plants. And the most common way of engineering a plant is through agrobacterium transformation of a plant, which is a bacterial to plant gene exchange. That's cross-kingdom gene transfer. And so this introduces a lot of uncertainty of where a gene can end up in the environment. And so as we do environmental synthetic biology work, that's so important that we are reducing carbon dioxide in the air, we're degrading plastic in the oceans, we're mining metals more sustainably. And as we move into more field trials and applied uses, I think we need to do so responsibly and consider where a gene that we introduce in the environment could end up. It's not all bad though. Like I said before, horizontal gene transfer is a very fundamental process that allows microbial communities to overcome difficult environments. And it's also a great way to engineer organisms to get desired functions. For example, researchers are using conjugation, which is a type of horizontal gene transfer that requires cell-to-cell contact to deliver genes to a microbial community in rhizobes, which is in plants, that can increase the rate of nitrogen fixation. This way, we're bio-augmenting genes that are already naturally present. Or we can introduce new genes into the environment that are better suited for our application. This way, we can greatly reduce the use of fertilizer needed to be able to feed our plants and feed ourselves, right? So in my thesis work, I focused a lot on this type of horizontal gene transfer, this conjugation. But there's still and we know a lot about the biochemistry of it, but there's still kind of a lot of open questions about how this works. For example, like the frequency of it, how often does conjugation happen? Or maybe the host range, and which organisms are participating in horizontal gene transfer in conjugation, or the dynamics. What path does a plasmid take? What order do organisms transfer genes in? Or how do our human impacts affect this process and all of these dynamic rates? So what strategies can I employ to understand a process that happens at a microbial scale? So there's a few strategies right now that we use to understand horizontal gene transfer, but they're a little bit limited for microbial communities. One of the most common is computational prediction. That's whenever you're looking at either a specific sequences within an organism that seem to be like suspiciously different. Or maybe, checking phylogenetic trees across each other and seeing maybe some gene seems to be more closely related to another organism than in the organism that it relates, that we found it in. And the issue with this is that, it's great to be able to detect horizontal gene transfer, but it has to be retained in a population long-term. So to be able to detect it, you have to actually show up in the database. You can only do it at evolutionary timescales because of this reason. And it's also difficult to find what is the donor species that actually sent in this gene, as well as depending on the type of horizontal gene transfer, it's difficult to see how it actually acquired that gene. Another type of method that we can use to understand horizontal gene transfer are experimental methods. For example, for conjugation, we use a filter paper assay where we're literally going to mix a bunch of microbes, put them on a filter paper, and then we can literally count which colonies have been transferred, right? Or we can use fluorescent reporters where a input signal is linked to a fluorescent protein that we can then visualize to see if something happened. This is how we've discovered a lot of the biochemistry about how these processes work. But often they require culturable microbes and ideal conditions. It's also really difficult to do this often in non-model organisms, just because a lot of different species have different processes, and it's difficult to make something work for everybody. And finally, often these require constant monitoring, especially with the fluorescent reporters, where you have to keep kind of looking at it to see when you see the spike in fluorescence. Another one output that's really useful though and that's very promising to understand gene transfer is genetic memory. So in this type of output, what we can do is we can literally write into the DNA of a cell whenever that cell experiences some sort of signal. So then we can read the signal much later to see when a cell experienced this. And this is great whenever we're studying complicated processes, or whenever we're looking at environments that is very difficult to get these visual reporters to work in. So for example, in soil, it's really hard to see a color change if it's like in the middle of the soil, right? Traditionally, we use proteins to write into DNA. And the issue is that these DNA writer proteins don't often work across all species, which makes them complicated to use in microbial communities. This is often because they either fold differently, maybe the promoters or the ribosome binding sites don't actually work in different organisms. Maybe they need specific conditions for that protein to work that are not representative of what these microbes experience. Maybe the DNA is not even accessible for this writer protein. There's kind of a lot of things that could be going on. So our team started looking beyond DNA and into RNA as something to encode information in. And what's really cool about RNA is that you can replace both the DNA writer protein as well as the DNA itself that you're encoding information in. Depending on your application, this has several advantages. So the first, it's portable. RNA kind of works across species. As long as it's expressed, it should work. It does. It is fast. You kind of skip a whole step. You don't have to actually translate into protein. You can just transcribe into RNA. And it is direct to consortia. You could technically put this in a microbial community in situ, and you don't actually need to be looking at it all the time in ideal conditions for it to work. For my synthetic biologists in the room, it's also tunable. You only need to adjust it at kind of the sequence level and the promoter level. You don't need to mess with RBSs, which can add an extra layer of complexity to all of this, as we have found. And it's also autonomous. This is really key. It doesn't actually need any helper proteins to work. And this is something that I think people don't talk too much about whenever you use Cas1, Cas2 proteins, is that you need a lot of helper proteins for that to actually function, which makes this nice to use. So now we need kind of two things. We need a pen, like a writer, to be able to encode, to write information. And we also need a paper, some sort of RNA that we can read out information from. For our writer protein, we chose cat RNA. That's a catalytic RNA. Cat RNA is a type of ribozyme that can basically conduct reactions. It'll splice in, and I'll talk a little bit more about that soon. And for our paper, we chose 16S RNA. Here's kind of a little schematic of it. Again, I'll go through this more deeply, but it is a piece of RNA that is conserved across all microbes, across a lot of species, that has conserved regions here in gray that we can look for this specific RNA, as well as variable regions here in different colors that allow us to find what species this actually belongs to, right? And so we use these to make RNA addressable memory, or RAM. So this is to record the information of DNA transfer in a microbial community. I'm going to abstract a little bit, and you can kind of think about the 16SR RNA as a passport and our technology, RAM, as a stamp. So a passport will record information about where its owner has been, but whenever they travel somewhere, it'll be stamped, right? And then later on, you can read out where its owner has traveled to by seeing all of the stamps in there. Similarly, the 16SR RNA, this is the E. coli 16SR RNA, is a massive kind of thing, has variable regions here shown in green that can identify their owner. This is kind of like the ID kind of page, as well as conserved regions where we can look for this passport in like the pile of papers, I guess. So what we can do is we can encode our cat RNA into a plasmid and then deliver it to a microbial community through conjugation. Once this plasmid enters the recipient cell, it'll express our cat RNA. And that cat RNA will find the passport, 16SR RNA, and it will literally like splice in a barcode. It'll stamp in a specific sequence that we can then look for. We can look for the unspliced 16SR RNA to see who is in this community, and then compare it to the barcoded 16SR RNA to see who received this plasmid. Yeah, we're good. Perfect. So what we ended up doing is we use this technology to understand wastewater microbial gene transfer. And so what we did is we made a cat RNA, we added it to a plasmid, we added the plasmid to a donor cell that is able to move this plasmid through conjugation. And then we added this to a community. So we went to a Houston wastewater treatment plant, we took a sample, we brought it back into the lab, and then we added our donor bacterium into this sample. We let it do its like conjugation thing. And then later on, we extracted all of the RNA in this community. We found the 16SR RNA to see who is in the community, as well as the barcoded 16SR RNA to see who received this plasmid, who participated in gene transfer. And this is our beautiful results. So what you're seeing here is a phylogenetic tree of all of the organisms that we found in our community, and highlighted in orange are all of the organisms that we saw transfer happening into. So we saw splicing happening. And here is our donor species right there. So just for our first design, we saw that RAM barcoded about, what is it, like 140 out of a 279 member community. This is like about 50%, which is really, really remarkable resolution onto who participated in this key process that is so hard to study. Another thing that we can do is we can kind of see, you can see that we can get who participate in gene transfer, even if they're not super abundant. So what do I mean by that? Here you'll see in the inner ring is the barcoded RNA that's in blue, and in the outer ring, it's the non-barcoded RNA. Like that's how much we saw, right? You'll see big patches here of gray where we didn't see any gene transfer happening. That is to be expected, especially conjugation often only happens between closely related microbes. And so, maybe that's what happened. But what's really remarkable is that we saw a lot of places, a lot of organisms where we didn't really see much or any like non-barcoded 16SR RNA, but we did see some barcoding. And you're like, whoa, that's kind of suspicious, right? But so how we're actually getting this RNA is we're using PCR to amplify something, right? So whenever you're amplifying just 16SR RNA that's not barcoded, you're biased towards stuff that is most abundant. So you're getting that kind of flooding in your signal. But if you're looking specifically for stuff that is barcoded, then you're biased towards stuff that is barcoded. So I think what we're actually seeing here are maybe less abundant members that are still key players in horizontal gene transfer and microbial communities. And that's a very, very powerful tool, because that means that we don't have to sequence so deep to be able to see these organisms. So I hope that I've convinced you that this is a very, very cool technology that we want to pursue. So where do I go in, right? So my project is focused on making these even better. So we saw a few like unbarcoded species, and I wanted to talk about why that is, right? So for us to be able to actually detect conjugation happening, there needs to be a few things that happen. First, that plasmid actually has to be delivered to a recipient cell. Second, that cat RNA needs to be expressed, so the promoter needs to be compatible with those species. And then that cat RNA needs to actually find its barcode to splice into it, right? And this last one is what I really want to go and tweak, right? I want to make sure that whenever we're lowering our false negative rate by making sure that our cat RNA can actually find a suitable passport for it to splice into. My second kind of motivation is I wanted to see if we could make selective cat RNA design. So what if I wanted to encode a type of information only in one set of microbes in a community? What if I just keep getting signal from these like highly abundant microbes, and I'm really just looking for other microbes. I don't want to get it flooded. I don't want to spend like thousands of dollars on NGS on my trash. So how can I control the taxonomic composition of the microbial community information that I am getting? So with that, I wanted to see, I had a hypothesis that I could leverage the natural variation of 16S RNA to control information storage in microbial communities. And for that, first, I made a program that can make universal cat RNA and see if it could work, if I can make designs for different kingdoms. Then I tweaked that program a little bit to be able to make selective cat RNA to get selective storage. And then finally, I did this in vivo to see if my computational pipeline has any real life validity. So let's go just dive into it. The program that I designed is called RiboDesigner. That stands for Ribozyme Designer. And it's a sequence-based program that can generate custom cat RNA designs for a set of organisms. So how this works is you'll feed it in 16S RNA from whatever your community of interest in, or whatever organism you want to target. And the computer will take that in, and then it will churn out a few cat RNA designs. And then you can use those for whatever experiment you want to do, and hopefully get as pretty data as we got. Right. Okay. So a few things about how this cat RNA works in more detail. I'll be using this kind of shorthand to talk about the 16S RNA. It's a huge molecule. So this is what this means. It's actually much bigger, but we'll be looking at specific sites inside of that we'll be targeting. So this is where our initial universal cat RNA actually targets. So then our cat RNA will actually find that target and then do its reaction. There's a few also key design parameters that my program takes into account to be able to design this cat RNA. First of all, you need a catalytic U. You need a uracil in your target sequence for this to work. This GU wobble pair is really essential for splicing efficiency, and you're going to get very poor splicing efficiency without this wobble pair here. So very important to have a uracil. Another thing that's very important is this U pentanucleotide. So these are 5 bases upstream of this U in the target 16S RNA. And again, these also need to be like really ideally perfectly complementary to those 5 bases downstream of this G in your cat RNA, or again, your splicing efficiency really tanks. Finally, we added the synthetic guide. This is upstream of the G in the cat RNA. It's about 50 bases. And we found that adding this here and having it basically be as close as possible to its 50 complementary bases helps a lot with, for the stability of it and with target binding, for it to actually be able to bind to your password, right? So in our first study, we did this very manually. So what we did is we literally aligned 5 species, and we looked for conserved sites, the conserved catalytic U's that we could splice into. And here, this is what I'm showing is all of these little handwritten notes are my by hand validation that we're actually doing this correctly. We ended up finding, I think, 4 or 5 designs that we tested, and then we chose our universal one, which is the one that's highlighted in green here. The other ones are highlighted in yellow. I never want to do this again. I want to be able to do this automatically, right? So I made a program that used kind of similar principles, but you can also feed in a lot more sequences instead of just 5 to be able to make our cat RNAs. So how it works is it'll take in a set of 16S RNA targets. It'll find every single uracil in there that could be good for splicing. Then it'll generate the complementary sequence to the U pentanucleotide of the ribozyme, and finally generate a guide. That way now we have a full ribozyme design that we can test against a set of 16S RNA targets and see how well it should work just computationally, right? So let's go through this step by step. The first thing that my program does is you'll feed it in a set of 16S RNA targets, just a folder of sequences, as well as a reference sequence. This is really important because I want everything to be in kind of the same numbering system. Whenever I'm looking for these catalytic U's, what I'm doing is I'm aligning each sequence with a pairwise alignment to the reference sequence. And I'm putting everything in the same kind of numbering system. So I'll say like, okay, this U happens at position like 89 in E. coli, in the E. coli 16S RNA. That way I'll have all of the sequences in my data set in the same numbering sequence, in the same numbering system, and then I can compare how conserved they are across my entire data set. Yeah. And then, so we have a bunch of U positions, and then we can calculate how conserved they are throughout my data set. So for example, here in the middle one in position 747, there are 2 out of 3 sequences have a uracil at that position. So that's like a, 2 out of 3 score. Is not a very well conserved catalytic U. However, in positions 89 and 1412, we see that there is a catalytic U, there is a uracil in all of our sequences. So those would be really good sites to look for whenever we're looking for a good place to splice into. Now that we found the catalytic U's, we can find the U pentanucleotide availability at each of these positions that seem to be promising. So you'll see here in the first one in position 89, there's 2 different motifs, right? Even though that U is conserved, there's actually kind of 2 subpopulations here with different motifs. And so I'm scoring each subpopulation based on how like abundant it is in my data set, basically. So even though the U is conserved, there's not a lot of very conserved U pentanucleotides. However, when I look at position 1412, for example, that exact U pentanucleotide motif happens in all 3 out of my 3 sequences. That would be a really, really good place for me to look at whenever I'm looking to make a cat RNA that can target multiple sequences. Okay. And what I did is I made a bacterial data set using the Silva database. I just took in a bunch of sequences. My bacterial data set is, 2,410 sequences, just to see, and it's equally distributed. I took in like a subset of I believe like orders that are not from the same genus to make sure that it's not like all E. coli in my bacterial data set. And then I wanted to see the distribution of these uracils on these sequences. So on the x-axis, this is the E. coli reference numbering index, as I told you, that is like, this is the position, this is what this position would be in E. coli. And on the y-axis is the coverage of the the catalytic U's. So we're higher, let's say if it's at 1, that means like 100% of my sequences have a uracil at that position. And we don't see really strong trends here. This is, I mean, it's just 1 nucleotide. But once we start looking at the U pentanucleotide availability, we start seeing these trends emerge. See here, it's the same x-axis, but here on the y-axis, I'm actually plotting the distinct number of U pentanucleotide motifs that I see. And in green are the variable regions in the 16S RNA. And you'll see that we see a lot more of these motifs, of these distinct motifs in these variable regions, and not a lot in the conserved regions, which is exactly what we would expect, right? So this is great kind of litmus test that I'm actually performing. My code is actually doing stuff that makes sense. And whenever we do a similar score as above, where we're calculating the U pentanucleotide coverage of every single one of these. So every single one of these dots here is a single motif at 1 position. And we kind of see that distribution as well. I also went ahead and scored how our initial design did in this bacterial data set. And we see that it actually scores quite well, which is not super surprising, right? Even though it's made with 5 sequences, it did splice into like 50% of our community, right? So it makes sense that it should be good in bacteria. I also went ahead and did a eukaryotic just scan. So here what I'm showing is the Escherichia, like Baker's yeast 18S rRNA, which is like the eukaryotic equivalent. And here the variable regions are different because there's different variable regions for eukaryotes. But still here you see very, very good match of the U pentanucleotide distribution with these variable regions, which again is exactly what we expect. And my eukaryotic data set is also, it's like 2,600 sequences-ish, right? And again, I'm plotting our universal design. We only did this with bacteria, so even though that U is present, that U pentanucleotide like does not exist in eukaryotes, which makes sense. We only use bacteria. But this is good to know, right? Because if we wanted to actually go into eukaryotes, we would have to design a new thing to be able to actually see signal. So good data. Hello. Okay. We also did this in archaea. So my archaeal data set is actually quite a bit smaller. And I'll tell you why that's a problem in a bit. But we do see kind of that trend that we saw where there's more U-pentanucleotide distribution across the variable regions, but we don't actually see that strong of a trend as before. I think that is because of my size of my data set. Whenever people submit archaeal, this is my hypothesis, but whenever people submit archaeal sequences to data sets, often it's like these like wild, like, new extremophiles that we don't know anything about. And this is like the only one of this subset of this taxa of it. So I think if I had a larger data set, I will be able to, find more designs and have something that's more comprehensive. But for now, just for a first pass, this is quite nice. And this is the M-Smithy 16S RNA. Okay, cool. So, and again, I have my original design here, which should, performs in archaea. It's fine. If we wanted to hit archaea, I think I would do this again with a larger data set. Anyway. Okay, now we found our catalytic U's and our U-pentanucleotides. Now I want to actually make the guide sequence like I talked about before. This is this sequence here. Like I said, it's like 50 bases. And what I'm doing is for every single U-pentanucleotide motif at a specific position, I'm getting all of the guide complements. That's this sequence here. That could happen. And I'm taking all of them here, and then I'm splitting them up into batches. And each batch, I'm doing a multiple sequence alignment and then like squashing it all together. And getting a consensus sequence for each batch. And then I'm scoring the consensus sequence based on how conserved it is. And I'm using this math. It's just a fraction. It's okay. But what I'm really doing here is I am measuring how confident I am in each base in this sequence. Basically, the fewer the number of possible bases there is, the better the score. And I'm using ambiguity codes to represent this. This is kind of like a little thing. So you'll see ATGC, everybody knows ATGC, that's what DNA is made out of. But there's also these like weird bases here. What these represent are like other, like, a little bit of like ambiguity, right? So bases here, ATGC, it can only be an A, T, or G, A, T, G, or C. And as we go lower in the scoring, the number of possible bases that we can be confident about increases. And so our confidence decreases, and that's reflected in this weighted score. And so what that means is that if we have a sequence that only has ATGC, then our score is going to be the highest possible, which is 1, because we're like, hey, we're really sure that this is a great guide sequence for our community. If it's all ends, which means we have no idea what the sequence is, it's literally nonsense, then it's going to have the lowest score possible, that's 0. Often we'll have something in between these, right? Where it'll have like a little bit of ambiguity in there that, so it'll score something in between 1 and 0. So now that we have guide sequences, what my program does is it takes in a set of test sequences and then evaluates our designs against them and looks for that position that it's targeting and then says, okay, at that position, how conserved is that catalytic U? How many of these sequences have a catalytic U at that position in my test sequences? Similarly, how many of these sequences have a U-pentanucleotide at that position that matches my design? Or finally, what is the average, like how good is my guide at actually targeting this? And how I'm doing that is I'm taking the guide sequence and doing another, I'm doing an alignment with every single other sequence there, and then doing a consensus sequence with this pairwise alignment, and then scoring that based on how ambiguous it is. So exciting, we're gonna have full designs. And when I do this, I see that they kind of look awful. And what I'm showing here is on the x-axis, that's the U-pentanucleotide score. That's how conserved that U-pentanucleotide is, or a U-pentanucleotide is. And on the y-axis, I'm showing the guide score. Ideally, we'd want stuff that are in this top right corner here. That would be amazing. And we don't see that. And I'm like, why? Why is this happening to me? Right? And the reason for this kind of like a big issue for primary design software for a lot of things, whenever you do multiple sequence alignments with a lot of different sequences, you'll invariably get gaps. These are real data that I generated. I wanted to make like a guide from 160 sequences. And when I do this full multiple sequence alignment, you'll see that there's just a bunch of different like gaps. And whenever people deal with this issue, often they'll like randomly generate different bases and then see how well this work. But I was thinking that we lose a lot of the sequence information if we're randomly just guessing, right? I wanted to take an approach that's more of an educated guess. So I was thinking, what is a optimization approach that I could use where I'm already using the data that I've generated? Because I've already made consensus sequences for my entire data set. And so I want an optimization method that's still relatively fast, that's still relatively fast, that's not going to crash my computer, and that maintains as much sequence information as possible so I can make this educated guess. And I want to see, could I use the rest of the consensus sequences to optimize to my existing guide design? So how I'm doing this is, again, this is the same figure as before. We chose the highest scoring sequence, but what I can do is I can look at the other sequences from the other batches and then align my highest scoring sequence with the other consensus sequences and look at positions where I see ambiguity. Here I've labeled in position 1, 2, 3. I'll look at the equivalent base and the other one, and I'll literally see if I find equivalent bases in between them that would lower the ambiguity. For example, in position 1, my highest scoring sequence has an H that stands for A, C, or T. But my other sequence has a T there. They both share a T, so I'm going to then replace that base at that position with a T. Similarly, at position 2, it's an H, the other one is a B. B stands for GCT, so they both share C and T. Y stands for C and T, so I'll replace it with that, lowering the ambiguity. And if I don't see any shared bases, I'll just keep the initial base as shown in position 3. Once I have this, I'll score it again, and then I'll get a higher score, hopefully. And I can do this over and over and over again until I either reach the highest score possible, 1, or I just don't have any consensus sequences left. So to take this approach then, so if I just did all of the multiple sequence elements of these 160 sequences, just like that, and did a consensus sequence, I end up having a guide that scores it's a pretty lousy guide. If I take this batching approach where I'm separating it to it and taking the highest scoring one, I get a higher guide score, but I also end up losing the context of all of the other sequences, right? But if I take this optimization approach, I kind of get the best of both worlds, where I can get good, I have all of the sequence context, but I'm also not being bogged down by all of this huge multiple sequence alignment, and I end up having a much higher guide score. Okay, so when I do this in my bacterial designs, I see that I've recovered my guide score, right? And again, my U-pentanucleotide score is on the x-axis, and my guide score is on the y-axis. And we see that whenever I don't optimize, it's kind of trash, but whenever we do optimize it, we do see recovery here. I also went ahead and plotted, again, our original universal design. And again, it scores quite highly, which is great, great news for us. But if we wanted to go further, there are a few other designs that we could actually target. We ended up just choosing this one just because it honestly scores pretty well enough, and it did seem kind of more incremental to test these. So whenever I'm talking about a universal design, I'm talking about, later in the presentation, I'm still talking about our original universal design. What is cool, though, is that my program also found better designs for the other kingdoms. So whenever I look at Archaea, I find that, again, our universal design isn't quite too good in Archaea, and it's really bad in eukaryotes. But so if we wanted to target these two kingdoms, there are quite a few other designs that we could choose. Okay, so I have then successfully made a program that can make better cat RNA designs. So it can leverage the fact that 16S RNA is conserved to find these good sequences, these good locations within the sequence that we can target. It'll then test each design against a test data set, which is great, to kind of evaluate the performance in silico. And then it found several better universal cat RNA designs, even though ours actually was quite good in the first place. But it also found better universal designs for all kingdoms too. So now that I have this program, can I actually tweak it a little bit to make selective cat RNA? And so just to remind you a little bit, what if I wanted to have a subset of a community? What if I really wanted to only target a little bit of a community? How can I do that? So I kind of took this approach. I Again, so how I'm testing my sequences, I get a set of test sequences. What I can do is I can test each design against two sets of sequences. I can test the design against a set of target sequences, which I do want to barcode, and then test that same design against a set of non-target sequences that I don't want to barcode. And then I can get those scores, say like the U-pentanucleotide score, and I can literally subtract those two scores to see how selective my design is. So this delta score here, the higher the score, the more selective it is, the better it should perform in our target sequences versus our non-target sequences. Yeah. So I did kind of like a little bit of a computational study on this. I revisited our initial wastewater microbial community in our initial study. And I looked at every single order that we saw present. And I want to see if I can make selective designs for each order. I made data sets taking the Silva database, like 16S RNA that is from these orders. And I made target data sets that only include one of the orders, and then non-target data sets that include all of the other orders and not that one. So for example, if I was going to use Pseudomonadellis, the target sequence data set should only have Pseudomonadellis order organisms, and the non-target should have all of these other ones and not Pseudomonadellis. And then I divided these data sets at different levels of taxonomy to see how kind of selective we can get based on how conserved the sequences are. Whenever I do this, say if I'm like looking for a design that only targets E. coli for some reason. And here on the x-axis is the U-pentanucleotide coverage. We see that as we go lower in the taxonomy, as all of that target data set gets more and more conserved, we see not only more good designs, but we see that the better designs are scoring higher. And that's great news. So now we have like a pipeline to make selective designs, and we kind of know a little bit about the design rules about this. So by evaluating any given design against two sets of sequences, some that we do want to target and some that we don't want to target, we can determine if there are any selective designs. And I've made this to make cat RNAs for several levels of taxonomy. And the more conserved our sequences are, the better, or the more selective our design can get. Yep. Okay, finally, I wanted to see if anything that I did actually like matters by testing this in vivo. Right. So for this, I went back to our original data set and I wanted to see, hey, can I target only Pseudomonadellis and not Enterobacterellus, right? So Pseudomonadellis and Enterobacterellus. So these are the most abundant species that we saw. These two are actually the same order in the Silva database. I'm just going to be talking, whenever I say Enterobacterellus, it's both of these two. And I made data sets would target Pseudomonadellis and not target Enterobacterellus. And these data sets are made out of 3,000 sequences of each of these orders, right? Again, that are not from the same genus, so we don't have a lot of the same kind of species in there. Right. And so I used RiboDesigner to make these selective cat RNAs, and I chose one. First, here, what I'm showing you is our original universal design, and I'm showing you here the catalytic U score here, the U-pentanucleotide score, and then the guide score for either Enterobacterellus, which we don't want to target with our selective design, and Pseudomonadellis, which we do want to target with our selective design. Whenever we look at our universal design, we see that it works pretty well in both. But whenever we look at my selective design that I generated, we see that U and that U-pentanucleotide does not appear almost anywhere in the sequences that I don't want to target, which is great news. The guide score is high, but the reason for that is whenever it's calculating a guide score, it's actually saying, okay, for every single sequence that does have a U in there, how good is that guide for that? So it doesn't actually take into account how many sequences have a guide here. So I don't think this is a great predictor of selectivity anyway, just because of how that score is calculated. Okay, and I did do some synthetic biology in this PhD. Most of it was not in this project, but I did do some cloning here. And this is our original universal cat RNA plasmid. And what I did is I first added a little barcode here. I just literally put two primers that are like facing other direction, and did a 1-part Golden Gate to add in a little barcode sequence that I can look for. And then I did a 2-part Golden Gate to replace our universal guide stuff with my selective design that I used polymerase chain assembly to make. And then what that gives us is kind of two plasmids, there's one on the top and the one on the bottom that we will be testing. One that is our universal cat RNA and one that is our selective, calling pcat RNA for Pseudomonadis. Yeah. So once we're doing in vivo studies, there are kind of a few things that we can target. First, so these are just the plasmids, but I wanted to see how much transcribed plasmid there actually is. I mean, transcribed cat RNA there actually is. And these are the primer binding sites in here. We're targeting this barcode sequence here. And then we also want to see the splice targets. So this is the E. coli, this is the general 16SR RNA. And I'm highlighting here where each of my cat RNAs are splicing into. So my pcat RNA is splicing into here, this region here, while my universal cat RNA is splicing into this region. And whenever we're looking for designs here, I chose these sets of primers that we can use to find just the splice product, right? So we're having the transcribed cat RNA, which basically has like all of the RNA that has a barcode in, as well as the spliced cat RNA that should only show the 16SR RNA that has been barcoded. And so Travis actually did these studies. He made like a beautiful PCR, which I'm very happy about. Whenever we look at just the transcribed cat RNA in two model organisms, E. coli, which is an Enterobacterialis, and P. putida, which is a Pseudomonadalis, we see that both of them have similar levels of transcription. That makes sense. They're both under the same promoter. Like there's going to be cat RNA there. But whenever we're looking at actually spliced cat RNA, we see that even though our universal design splices into both of our model organisms, our selected design, the selected design that I made, actually only splices into P. putida. It shows remarkable selectivity for Pseudomonadis, which is wonderful, wonderful news. And so I think that's pretty convincing data that we can use this kind of sequence-based design to be able to make selective cat RNA storage, at least in vivo. And the guide score is not as useful as for this specific use case, but the U-pentanucleotide coverage seems to be a very, very good metric to determine selectivity. So what kind of can be done? I've talked a lot about what RiboDesigner has done, but what could RiboDesigner do? So first of all, we could use it in other environments. We could use it like to study full communities. What if we wanted to look at like the bayou water or like the compost pile or like effluent or the ocean? What's gene transfer like in there? Maybe we can do that. It actually doesn't need 16SR RNA. Like my program doesn't know that it's doing 16SR RNA, right? What it only knows is that it's aligning to another sequence. So as long as you're giving it a conserved sequence, you can really target other sequences that are also conserved. Some possible recommendations would be RPOB. I think it's an RNA polymerase near S, a nitrite reductase, and REC-A, a DNA binding protein. These are often used as like replacements to 16SR RNA for different purposes. These could be good targets. And finally, can we do this in eukaryotes, right? So actually my undergrad, Ashwin, is trying to do this in yeast, which is very exciting. There's that, eukaryotes are a lot more complex than bacteria. There's like a lot of other like junk in there. Like that makes it a little bit more difficult. But I made a set of yeast-specific designs for him because we want to make sure that whenever we don't get signal, it's because we have to optimize something else other than our design. But in summary, so what our team has done is we've created a new type of memory, which is RAM, that uses RNA as both the writer and the paper to encode information about a community. It's a new addition to the SynBio toolbox that can complement these DNA-based memory systems for various application in complex communities. And we've used this to understand the host range of some plasmids encoding this cat RNA. What I've done is I've made a Python program that can make better universal ribozymes that target most of the community, as well as selective cat RNA that can target specific community members while purposely avoiding others. Using this design, I made a selective design and I tested it against 3,000 sequences from 2 orders. So Pseudomonadis are the most abundant orders that we found in our wastewater. And we found that even though similar levels of our universal and selective ribozyme were transcribed in vivo, There's two metrics, this catalytic U and the U pentanucleotide availability, were highly predictive for how well these would work in our model bacteria, with our selective design only splicing into P. putida and not into E. coli. So with this program, I hope to allow anybody from, to use our technology to understand more about their microbial communities. And I'm really grateful for the opportunity to show this to y'all, and I'm excited to see what applications you come up with. And so I would like to thank my lab, honestly. These are my funding sources here. Thank you for giving me money. And 5.5 years is a really long time. A lot can happen in that amount of time. We went through a whole pandemic and out of it, and it was really rough at times, but through it all, I had so many people supporting me throughout. And so I want to first thank, if it clicks, I want to first thank my labs. I've enjoyed all the dinners, all the piercing parties, all the white elephants. I've loved our white elephant parties where I get to steal things from Lauren's children. My citizenship party, I got my citizenship while I was here. That's how long I've been here. And they threw me a really lovely party. I really appreciate all of your support. I really want to say a special thank you to Joff and Lauren. Actually on the top left corner, that's a cupcake that Lauren got me during COVID for my birthday. Both of you have been so supportive for me during this whole journey, being so accommodating, so patient with me when things got really rough. And I am really, really so grateful for all the support and for cheering me on and helping me feel capable when I couldn't. And also thank you for letting me go to a bunch of conferences. I've met a lot of different people. I've had so many wonderful and weird experiences. And for the conferences that I've had the privilege of attending. I want to thank my friends. This is my friends from Rice, from Culver, my online friends. We've had so many adventures. We had so much good food. We've made mooncakes. We've eaten. We've stolen corn. It's been really, really lovely. So many foods, so many workout parties. I really could not have done this without you. I want to thank my family. My grandparents and aunts and uncle who are on this Zoom. Thank you. Pato and Pepe, who have shown us resilience and what a loving relationship can look like. And my brother, who in the span of my PhD graduated from both undergrad and cooking school. And I want to say a really special thank you as well to my dad for making the effort to always come here for Houston, so that we can eat at all the wonderful places here, and for pretending that it's his birthday so we can get extra dessert at every single place we go to. My mother-in-law, who has really accepted me into her family with open arms. I'm really, really grateful. From the first day that we met, you've just welcomed me. And my mom, who is really my inspiration, just like the woman that I want to be. She's taught me everything about everything basically. Muchas gracias, mamá. I want to thank my girls, especially, so B and Brooke were essential in writing my thesis in this presentation. They were very important. I want to dedicate this to Hazel, who passed during our time here, and to Rosita, who without her, I would not be here. And then finally, I would like to thank Cole, who's my favorite person, who's my partner. We've been together for such a long time, and I've been so privileged to be able to have you by my side through all of this. And you've empowered me, you've taken care of me during, really rough times and helped me through my disability. And even you still managed to be an outstanding scientist and do your own PhD through all this. And I'm so proud of what you've accomplished, and thank you so much for being with me. And with that, I also want to thank all of you on Zoom for being here, all of you here who are here physically. Thank you so much for being here, and I'm happy to take any questions. Cool. And on Zoom, I'm also filtering questions. So if you want to say that, I will just read out the question. Yeah. Any question? Yes.
Kiara, I know you have done a lot of really complicated code in your time here.
Yeah.
It also means a lot of troubleshooting.
Yes.
What troubleshooting are you most proud of?
Oh, what troubleshooting am I most proud of?
Yes.
Oof. Actually, when I first started this program, What I was doing is, so the reason I'm making kind of like a big deal of this alignment here, it's because the first thing I did is I just took in like all of these sequences and just found for every single U-pentanucleotide motif like possible in the sequence, and then that ends up being a really, like, the more sequences you get, the most horrible like your computational steps get. And it took me, I went from like knowing no coding to like generating like some really complex, silly thing to be able to deal that. Like it was like multiplexing, and then going back and kind of thinking, hey, do I actually really need to do this? And going back and like simplifying it. So I'm pretty proud of like, being able to like kind of step back, think of it, and then use the things that I've learned to better my code. Yeah, thank you. Anybody else? Yeah.
Great job.
Thank you.
So it seems like one of the first things you need to do in your pipeline is like choose a reference sequence.
Yes.
So I was wondering, how do you think your choice of reference sequence, like maybe it could affect, your outputs from there?
Yeah, so I did choose like different reference sequences. Mostly it's to get everything in like the same like position. It doesn't actually change it too much. Some things will be called, like let's say if you use like Pseudomonas instead of E. coli. Some things will be called in other positions instead of that. That mostly affects like the variable regions, but the stuff that's conserved is often still pretty conserved. My program actually, if you don't actually give it a reference sequence, it'll just randomly select one from the data set to be able to deal with that. But just because 16S is so conserved, I didn't see any huge trends. Yeah. Any other questions? Yeah.
So at the start of your talk, you talked a lot about horizontal data transfer.
Yes.
Got it. Have you seen anything about horizontal data transfer in RTLs that like?
I personally haven't. I haven't really looked at it honestly, just because I think even like the existence of Archaea sometimes, depending on the day, is a hot topic of contention. But I mean, my kind of thing is like it just kind of happens everywhere, right? Because even if the bacterium is dead, if like even if the DNA is free floating, it can still be incorporated into something else. So, and the other thing is again, like the kind of systems that we have set out to understand these kind of dynamics are really like biased and poor. So even if I did see that, I wouldn't really trust like specific rates. But I can feel confident saying that it is happening, like for sure, somehow. Yeah. Travis, hello.
Where can we access this code?
Yes, it is right now. It's on GitHub. I'm cleaning it up. So it's not public yet. But once it's like we're planning on getting this paper out like ASAP. And after that, I'll use it and then scream at me for my poor code annotation. But I'm annotating it. But yeah, it should be out soon. Yeah.
Well, this ends the public portion of the defense. Like, thanks,
Oh.
Kiara, again.
Okay. Oh, and Tasha, I do see an application in the gut microbiome, but thank you all for being here. Okay, I will see y'all soon. Bye.